Hello friends, today I'll be discussing a question on the topic replication. One of the questions which has been asked is outline the steps of replication. Now I've given this steps of replication in points, and depending on the weightage of the question, that is depending on how much marks has been assigned to the question, you can elaborate on it and pick up extra points from the textbook. But this discussion will give you exactly how to attempt this question. What are the main points that you should write when you are discussing the steps of replication? Now you all know that the three stages of replication is initiation, elongation, and termination. Initiation means the unbinding of the two complementary strands. Now this unbinding occurs at a specific site. This site is known as the origin of replication or the ORI site. At this point, you have the formation of a replication fork, and the replication is bidirectional. Now, what are the steps of initiation? Firstly, DNA A protein will recognize, bind to the ORIC site and denature the DNA. Second, DNA B protein or helicase will bind, unbind and form the replicating fork. Third, if there is any torsional stress which is formed because of this process of separation of the two strands, this is removed by the topoisomerases. Fourth, single-stranded binding protein will bind to the separated strands and stabilize the two strands. Fifth, we have the synthesis of the RNA primer. Now the next stage of replication is elongation. DNA polymerase III will initiate the synthesis of a new DNA strand in the 5' to 3' direction. Now it always adds deoxynucleotides to the 3' end of the primer which has been synthesized. Now both the strands are synthesized simultaneously, but it occurs in opposite directions. The leading strand synthesis occurs towards the replication fork, whereas the lagging strand synthesis occurs away from the replication fork. The leading strand synthesis is always continuous, whereas the lagging strand synthesis is discontinuous, requiring a number of RNA primers. Each of these RNA primers are extended by DNA polymerase III into short pieces of DNA called Okazaki fragments. Now, after the lagging strand synthesis is over, the RNA primers are removed and the gaps are filled by DNA polymerase I. Nicks are then sealed by DNA ligases. Termination, the third phase. Termination sequence or the TER sequence will direct termination. So there are specific proteins known as the TER binding protein which binds to these termination sequences and prevents the helicases from further unbinding the DNA strands. And these sequences now facilitate termination. After termination or during the process of replication, proofreading and correction of mismatches occur, particularly with the help of DNA polymerase I and DNA polymerase II. This diagram is important and has to be put in if you are asked a question on replication. It gives you it depicts how the two strands are separated. The two strands are separated with the help of the enzyme helicase. Torsional stress is removed with the help of the enzyme topoisomerase. Single strand binding protein binds to the two strands and keep the two strands separated from each other. A DNA primase will force the production of numerous RNA primers. These DNA polymerase I then adds on nucleotides to the RNA primers. Okay, and once the lagging strand synthesis is completed, the RNA primers are removed and the gaps filled by DNA polymerase 1, and any nicks are sealed by the DNA ligases. In the case of leading strand synthesis, it is a continuous process. There is no formation of Okazaki fragments. Okay, so this gives you an idea of how to attempt the question on the steps of replication. Thank you.